Hello, everyone, and welcome to today's webinar, Evolution of Scale for Synthetic Oligos. I am Matt Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. All questions submitted today will be answered via email following the broadcast. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. There will be several polls distributed throughout the webinar. Your participation is greatly appreciated. I'd like to now welcome our speaker, Ben Hinton, Senior Operations Manager, Genetic Science Division, Life Science Group, Thermo Fisher Scientific. Ben, you may now begin your presentation.
Okay, hello everybody, and welcome to the webinar on the evolution of scale for oligonucleotides. Today, I'm going to cover six topics. One is who we are as a company. The second is the evolution of scale from oligonucleotide synthesis. Third one, oligonucleotide synthesis from discovery work to commercialization, followed by small-scale oligonucleotides and their applications, and then large-scale oligonucleotides, their applications, and then why scale-up matters. So, who are we? Thermo Fisher Scientific is an instrument and consumable company who have around about 130,000 employees throughout the globe. We have a vision to enable our customers to make the world a healthier, cleaner, and safer place. Within Thermo Fisher, we have 27 years of experience in oligonucleotide manufacture, covering all of the different types of oligos, primers, probes, and gene synthesis, all built to customer specifications. We also have an Oligo Center of Excellence within Pleasanton, California, and we are accredited to ISO 9001 and 13485. My name is Ben. I'm the Senior Manager for Manufacturing Operations within the Pleasanton site. I have 24 years of experience working for Thermo Fisher, spanning various oligo manufacturing sites within the UK and within the US. I currently lead the specialty oligo and custom development and synthesis lab at Pleasanton. And the next topic is the evolution of scale for oligonucleotide synthesis. So in recent years, synthetic DNA oligonucleotides have enabled novel applications with development in synthesis technologies for large-scale oligos. From this perspective, the webinar will briefly review the techniques and technologies that enable the synthesis of large-scale oligos, the challenges that arise with an increase in scale, and emerging applications from R&D to clinical diagnostics and then therapeutics. There are three main learning objectives throughout this webinar. We want to understand oligosynthesis from discovery work to commercialization, help to identify those technical complexities faced during the scale-up of sequences for our customers, and then make sure that you, as our customers, know what services and support opportunities Thermo Fisher Scientific can provide to you and your company. So let's start at the beginning. So what is oligonucleotide synthesis and how does it work? So oligonucleotide synthesis hasn't really changed much in the last 30 years. We still use phosphoramidite chemistry. We start off with a solid support that's functionalized with the first base or modification of the sequence. And then we remove the first DMT group or blocking group from that functionalized support, which enables us to add the next base with an activator to that functionalized site that we've just deblocked. Obviously, this reaction is not 100%. It's about 98% efficient, and so there are those shorter sequences that do not get the next base added. So the next step after activation is capping, where we cap those unreacted sites to prevent further extension of those chains. After we've capped those non-reacted sites, we then oxidize the tri-ester bond to create a stable phosphate bond, which will allow us to then repeat the process of deblocking that added amidite. And then we just repeat that cycle over and over again as we add each base to the the chain and sequence that has been added. So like I said, there's no real difference between the chemistry that we've been using for the last 30-40 years. And so small scale and large scale synthesis both go through that same phosphoamidite chemistry. The difference really between those two small scale processes is really how do we manage those different scale of samples, and what challenges come when we scale up from a small scale synthesis to a large scale synthesis, and how do we manage through those processes. These oligos are also used in varying applications, such as PCR and target identification for diagnostic work, therapeutics and aptamers for more clinical trials and therapeutics. So, question: what scale of oligos do you currently work with? Okay, so on this slide, I just want to talk a little bit about small-scale oligonucleotides and applications. And small scale, what is small scale? So when we talk about small-scale synthesis, we're really talking about any synthesis that is less than 10 micromole scale. It's a manageable scale, and the process is very robust, and we can make it on a lot of high-throughput platforms. We can make multiple different oligos at a time, up to 197 oligos on one synthesized platform. So we are consistent, repeatable. We also include basic modifications and purifications as needed for these small scale oligos. And they have, again, varying applications. So one of the main applications that we would use these oligos for is diagnostic testing kits. So we would create a probe with a quencher and a fluorescent dye for a specific target sequence within, for example, a COVID virus. We would then have the forward and the reverse primer that is amplifying that section where the probe sits, and as that amplification occurs, the probe will be consumed by the extension of the primers, and the FAM molecule will float away and start to fluoresce. And it's that fluorescence that gives you the signal that there is that target sequence within your sample. So this PCR reaction, this endpoint PCR, can be used for multiple different applications throughout lots of different sectors of the market. Typically, a small scale synthesis will provide a lot of different reactions for these types of PCR reactions. So, for example, 25 nanomole synthesis scale delivers around about 1,000 PCR reactions. So you don't really need a lot of material to get a lot of tests out of a small scale synthesis. So that's one of the benefits of small scale synthesis, is you get a lot for the scale that you make it at. But it doesn't always work depending on the application that you're trying to design for. Small scale synthesis is also very, very established. So we are not really worried about the quality and the consistency of the products made on this process. So we spend a lot of time looking at primer design and analysis versus whether or not the oligo is going to be of a good quality. And obviously the same synthesis processes can aid in the development and the R&D that goes into these oligotherapies, which require a larger volume of oligo, but for feasibility studies, they are ideal. And so we would start with a feasibility at a small scale and then progress within scales as we progress through the workflow. Large-scale oligonucleotides are a completely different process. For us to make a large-scale oligo, we are really talking about 100 grams of delivered yield. And this causes a lot of increase in complexity. We can think about how for a large-scale synthesis, it's not only the design that is really important, but also how we manufacture that oligo and how much sample that we have to deal with and how we deal with those. So for example, to desalt a small scale oligo, it's really simple. We're only really dealing with 1 milliliter of sample. We can really easily ethanol precipitate that sample to remove the salts. But when you've got 20 liters of sample, we can't just do the same process. So we have to think about something different. And so what we do in Thermo Fisher is we have multiple different processes that we can use to desalt an oligo. And as we go through the development process for large scale synthesis, we get to choose what type of desalt or what type of purification really suits the application that the customer wants us to use. So we have a lot of knowledge and a lot of experience in making those types of decisions as we go through the synthesis and the process with our customers. Applications for large-scale oligos are usually therapeutics. So we have antisense oligos that are made in the majority of therapies. These are oligos that are short and have the ability to modify RNA and reduce and modify protein expression through several distinct mechanisms by targeting the source of the pathogen as opposed to the effects of the pathogen later in the downstream pathway. We also make aptamers, which are synthetic antibodies and are very, very specific to the antigen that it has been designed to bind to. So why does scale up matter? Well, as I've said, there are a lot of complexities and technical issues that we have when scaling from small scale all the way up to large scale. And what that translates to from a customer's perspective is that customers sometimes do not have the internal technical knowledge to understand why things can be different when they're looking at a feasibility study using a small scale oligo to a therapeutic batch size of large scale oligo. So that is something that we're very used to working through as a company. And we also can sit down with our customers, talk about the capacities and the timeline of doing these scaling efforts from small scale to gram yields for their application. And also sometimes understanding of those regulatory requirements is difficult to get to because we're looking at what those therapeutics are used for. So do we need endotoxin testing and do we need bioburden analysis? So these are those conversations that we can have with our customers to decide which is the best process to use as we scale up. And that's where the solution comes through. So you really need a dedicated technical project team to really be able to answer those questions that you have and to be able to explain the differences between the small scale and the large scale and explain why we need to do things a different way and how we're going to do that. We also make sure that we are, governed by the ISO standards. We are 13485 registered, and we are able to get that repeatability that our customers need. And we also do analytical analysis on all of our lots, depending on the customer requirements as well. So for the next couple of slides, I'm just going to talk through some case studies that we've had in the last few years. I just want to use these to highlight some of those technical complexities that we can encounter when we are deciding to scale up our sequences. Excuse me. So the first case study, one of our customers needed a really complex backbone modified oligo, which included very different modifications, LNA bases, a photocleavable spacer, and they wanted really high purity of above 90% with various different fluorescent dye attachments. Obviously, these oligos are very complex, and the scale-up method was something that we had to really think about. How do we do what we're being asked to do? So we had a partnership with the customer. Within a few weeks, we'd sat down, we'd talked and discussed with the customer what their actual needs were. I'd worked with our manufacturing sciences team and our development team to come up with a process that was going to deliver exactly what the requirements were. And within a few weeks of establishing that, the client received their first milligram of product. We sent that to the customer so that they could test and make sure that the quality that they needed was fit for their purpose. And they came back and said that it was very good quality, superior to quality they were getting before. And then they placed some more pilot orders for some other samples and for some other scales to try and assess what scale they really needed in order to satisfy their demand and their forecast going forward. So one of the benefits that conversation and that partnership that we had with the customer was that we could really get down to what specifications did our customers really require and what were unique for us to meet that project's needs. We set out a very clear delivery timeline, which meant that we were able to meet that, and our customers knew exactly when they were going to get their oligos for testing, and then when they were going to be able to get the next pilot order for them to work on as well. And we also were able to set up a long-term pricing model because we did have a forecast from the customer, so we were able to understand what the requirements going forward were and how that would affect our capacity within the lab. So the outcome of this particular order and this particular partnership was that the client was able to bring their technology to market on time and well within their budget. So that was a real good example of when working together closely with the customer really paid dividends, both for our team here in Pleasanton and also for the customer as well. Another case study. This is a biotech customer that needed a really large scale, complex, GC-rich, unmodified sequence with really high purity, and a lot of analytical testing after the fact. So the client really wanted to ensure that we could deliver an oligo that was ready for their analytical testing within a really tight timeline. We did have some technical issues with this oligo. It was very G-rich and the scale was very, very high. So we started off making around about 1 to 2 grams of the oligo. And we did that with the pilot builds within, I think, 3 weeks, and satisfied all of the purity requirements that the customer wanted and all of the analytical testing that was required, and delivered that to the customer. They were happy with that and came back for a higher scale. So we needed then to scale up to 10 grams per build. And when we started to do this, we came across quite a few technical issues. So the G-rich oligo had a secondary structure that really became prevalent as the scale increased. And so we had to change our process to adapt to how we were going to resolve the purification problems that came with having a secondary structure on such a large scale oligo. So we changed the synthesis strategy. We adapted the purification method so that we could purify a trityl on oligo. And then we had to introduce another couple of steps to remove trityl and then to desalt that oligo using ultrafiltration. But those changes were communicated to the customer. We talked through the reasons why we had to make those changes, and then implemented them for the large scale 10 gram build, which was successful. And then we moved on and now have a process in place that allows us to deliver those oligos and those types of oligos regularly and relatively straightforwardly now that the process is in place. So again, another benefit of working very closely with the customer is that we get to know exactly what those customers need from a specification point of view. So another thing that this customer specifically asked for was bioburden testing, ash content analysis, and heavy metal analysis, which as a company we hadn't done before. So we then reached out to other vendors and found another third-party vendor that could carry out those tests for us so that we could take it off our customers' hands. So before we even ship to the customer, we make sure that we meet all of those testing criteria for their oligo. We take all of that burden off the customer and we take it in-house, and then we make sure that we're delivering what they need. And the outcome of that is that the client scaled up to multi-gram lots for their application, and then came back to us with multiple other sequences that they wanted us to scale up as well. So, would you like to connect with our technical team to learn more about our capabilities for oligo synthesis scale-up? Okay, so in order for us to help our customers with the scale-up, we have put together a checklist. I'm going to walk through that now because it's very important, and it really does help you as the customer and us as the manufacturer to make sure that we're delivering exactly what you guys need. So the first thing is you need to have the right support. We have technical project teams that are skilled in oligo development and capacity management. So that team will reach out and collect the requirements from the customer and do an initial technical assessment. If we feel that we have to reach out to the customer and sit down with them and go through step by step, then we will do. If we feel that we are in a place where we can put a quote together with those specifications, then we can send a quote and discuss with the customer afterwards if needed. We need to make sure that all those quality systems are in place within all development and production labs, and that any pilot order that we do manufacture for our client is manufactured under those quality systems and properly documented. We then would send the customer the oligo for testing, and if that goes successfully, then we would continue to provide the customer with their required material. And if not, then we still have this feedback loop where we can go back and we can discuss any technical challenges that the customer sees or that we've had throughout manufacture and make changes if need. So development really is an iterative process, and flexibility on both sides is necessary for the scale-up. After we've gone through that whole process, then the scale-up is in place and we're able to provide the material that our customer needs. So, Thermo Fisher Scientific Services and Support. We do have a lot of support functions that are available to our customers. We have very, very strong technical teams that are able to jump on a call, answer emails and answer questions that our customers have. We have 100 years of combined experience throughout the team to draw upon, and we are able to make anywhere from a couple of milligrams of oligo all the way up to 100-gram capacity, really. We have a dedicated ordering and fulfillment service that are integrated with your systems and processes. We have ISO 9001 and ISO 13485 compliance quality management systems. And as I said before, customer service and technical support teams that are there to help you with your needs and requirements. Would you like a quote on your custom oligo design? Okay, so are you ready to get started? Are you interested in having a conversation with our technical team? Below are 3 easy steps that have been listed to make it easy for you to reach out to our technical teams and start your technical evaluation. Step 1, make sure that you have all of your specifications and technical requirements, such as delivered yield, purity, modifications, etc., with you. And then reach out to our small scale oligo team. If it's just a standard oligo, you can order them at the links below. An inquiries email address that is referenced there, and one of our technical project managers will reach out to you within 1 to 2 business days to start your project kickoff. The third step is that your technical project manager will work with our manufacturing sciences team to determine feasibility, timeline, and the price. They will send you a comprehensive quote and work within your budget and timeline. Any questions today? And thank you all for listening.
Thank you, Ben, for your informative presentation and your time today. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Questions submitted today and during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.